Hello, everybody. Very good evening to you all. Good evening, everyone. So, as you all know, CSIR exam is merely 2 weeks
Hello,
away.
everybody. Very good evening to you all.
So, CSIR exam, as I was telling you, is just 2 weeks away. And I hope that all of your preparations are going on in very, very speedy way. And I hope that all of you are doing enough of question practice and revision. So, guys, with much students' demand, actually, we are bringing back Brahmastra series. And starting from today, every day, you are going to have 1 hour of Part C question discussion from every unit, starting from today itself, 6 to 7 every day in Biotechnica's YouTube channel. So, without further delay, let's get started. So, today we are going to talk about, myself, Tithi, and as you know, I will usually deal with Unit 8 genetics. So, today I have got questions, especially the questions from the previous year CSI papers, especially those type of questions which has been asked repeatedly from the subunits F to K. Okay, so let's cover this 5 subunits and see the important questions or the type of questions which usually come from there. So let's get started, everyone. And I would definitely like to have an interactive session. So if you guys have any question, please feel free to drop a message in our chat box. Okay, let's get started, guys. So this is the first question which is from CSIR NET December 2023. Okay, the last exam itself. This question says that Drosophila male carrying an X-linked temperature-sensitive recessive mutation that is lethal at 29 degrees Celsius but viable at 18 degrees Celsius. Okay. Now, what is it saying? That it is talking about a X-linked temperature-sensitive recessive mutation. Now, because it's a recessive mutation, so just assume that suppose this is caused by a gene X, okay, which is X-linked, of course. So let's say that capital X will be the dominant allele and it will be normal, whereas small x will be the recessive allele and that is the mutant. Okay. Now this X small x is a temperature sensitive mutant, okay, which is lethal at 29 degrees Celsius. At 29 degrees Celsius, it is lethal. Okay, I'm writing L. At 18 degrees Celsius, it is normal. Okay, now all this information, if you see, that is only given to confuse you. Okay, because here in the last part what it says, that if the eggs are laid in both the cases and are reared at night 29 degrees Celsius, what will be the male female ratio in the given progeny? So because it is 29 degrees Celsius only, there is no complication, so you have to consider that small x is lethal. Okay, so there is no complication of small x also being normal. That is completely ruled out, right? So this is only to make you confused. Right, now it is mated. So first of all, we have the male. Okay, so what will be male's genotype? So I'm writing it here. So the male's genotype is small x and y, right? It carries a X-linked temperature sensitive recessive mutation. Okay, and this is Drosophila, remember that also. Now, when this male is mated with a normal female, okay, so here it is normal female. So, normal females genotype will be capital X, capital X. Okay, capital X, because she doesn't have this particular mutation. It is not mentioned that she is carrier or anything. She doesn't have it. Okay, so in this particular case, so for A, if I say, so this is our male for both A and B scenario. So for A, female is capital X, capital X. Okay, so what will be the cross? Cross will be small x y crossed with capital X, capital X. Okay, now you can make a Punnett square, very, very simple. Okay, there's actually no need of making this second one also, because she is homozygous. So both the she will produce only one type of gamete with the capital X allele. Okay, now in this situation, what do we see? So all the females will be carriers. right? And all the males will be normal. Okay, so what will be the question is, what will be the male female ratio? So in this particular cross, male is to female will be how much? 1 is to 1. Okay, now once you have identified this, all right, you have got your answer actually. Option 2. Okay, so if you just do this half a question also, you can get your correct answer. Okay, but let's see the second one, and actually the second one is the complicated part. Okay, and many people will get confused in that, but you don't have to be confused here. Okay, so let's have a look at the second situation. So B says that a female containing attached X chromosome. Now, what is attached X chromosome? So, in attached X chromosome, so basically there are two X. This is how we write the attached X chromosome, where two X chromosomes are joined with each other. Since they are joined with each other, they will always be inherited together. Okay, so the female here will be attached X chromosome, but now remember this is Drosophila. Okay, so in Drosophila, sex determination is based on X is to A ratio, right? So here it will be so the female here will be this female genotype is this. Okay, that she will have a pair of attached X chromosome and another will be a Y chromosome. Okay, that is what the female with an attached X chromosome genotype will be. Okay, now once you have got it, now in the same way as we created the cross earlier, so here also the cross will be done. So this individual will produce two kinds of gametes. This individual will also produce two kinds of gametes. Okay, so you can make a Punnett square here. Small x, capital Y. Okay, so what will be the genotype in this box? Attached X chromosome and the other one small x. Okay, next one small x, Y. Here attached X chromosome and Y, and here Y and Y. Okay, now in this situation there are a few things. Okay, first of all, this portion and this portion, okay, which are here they have 3 X chromosomes and here it is 2 Y chromosomes. So both of this progeny will be dead. Okay, so this will be dead even when there was no temperature sensitive lethal mutation also involved. Okay, so this will be dead anyways. This two proportion progenies or the proportion of progenies will be dead. Okay, now what about this part? This part will also be dead. Why? Because of the X-linked recessive mutation. Okay, because the embryo will be reared at 29 degrees Celsius, in which the mutation acts as a lethal mutation, and this individual, this part of the progeny will also be dead. Okay, so who is alive? Only this part. Okay, and for this particular part, so this progeny is alive, right? And what will be the gender of this progeny? So again, in Drosophila, because it is X is to A ratio, which determines the gender. So here it will be here, because it is 2 X chromosomes are present, right? So it will be a female. Okay, so this will be only female. B will be only female. Okay, so you have got your answer. So as you can see, you can get your answer only with doing the easier part also. Okay, but many times students will do what? They will see this part, they will think that I don't know what it is, and they will just skip the question. Okay, so you have to practice this kind of questions and understand how to proceed. Moving on to another question that also from last year, December 2023. And as I tell you in the class all the time, that this is a type of question which has been asked multiple times. Okay. So if you see previous year papers, like if you can take last 15 years papers, also this type of question has been asked. So here the question says that a figure given below is of two homologous chromosomes paired during meiosis, where one event of recombination occurred between two homologs. The following interpretations were made. So there are statements given A, B, C, D, E. Which of the following options have all correct answer? Okay. So the first one says that the individual is heterozygous for an inversion. Okay. Now let's see whether it is correct or not. Yes, it is true. How do we know that? Because only one homologous chromosome is making this cross loop. Okay, so if both the chromosomes or both the homologous chromosomes were carrying the inversion, then there won't be any problem in aligning, right? They can align just like the normal homologous chromosomes. But because one of the homologous chromosome is making this cross loop, that means that it is heterozygous for the inversion. So A, correct. Now B, the figure depicts a paracentric inversion. So for that, what we have to do, we have to look into the loop. Okay, so whether the loop includes centromere or not. So as you can see that the loop does not include centromere. Centromere is coming outside. Okay, so that means inverted segment do not include centromere, and if it does not include centromere, then it is called as a paracentric inversion. Okay, again, correct. Now for statement C, after recombination at anaphase 1, a dicentric and an acentric chromosome will be formed. Now that is absolutely correct. That will be the consequence when there is crossing over at this point, right? So this will join with here and it will result in the formation of a dicentric chromatid. The other chromatid which will be formed from here will be an acentric chromatid. Okay, so statement C also correct. Now, at anaphase 2, the recombinant chromatids will have large deletions and duplications. Now, there is a minor mistake here. So, in this case, this statement D should be wrong because in case of paracentric inversion, the recombinant gametes will be inviable, but that will be because of deletion alone. Okay, because this acentric chromatid gets completely deleted. So because of that, many of the genes will get deleted. So the recombinant gametes will not receive a single copy of many genes, because of which it becomes inviable. Okay, so it says that because of large deletion or duplication. So duplication does not happen here. Duplication is correct for pericentric inversion. Okay, so as of now, let me say that this statement is wrong. Okay, now why I am saying, because I'll show you the options. Okay, now statement E says that inversions are often considered as crossover suppressor because crossover product does not survive. Now, this is also true. Okay, so according to our calculation, what do we see? Which statements are true? A, B, C, and E. Okay, now when you scan through the options, you will see that A, B, there is no option with this A, B, C, and E. Okay, but you have an option which includes all of these statements along with D as well. Okay, so your best guess will be to choose that option, okay, which comprises of maximum of the correct choices. All right, so that is the reason why I took this question. So this is the second reason. So many a times it happens, so answer will be option 4, okay, according to the option given. Otherwise, if there is an option of A, B, C, and E, that would have been more correct, because duplication does not duplication of genes or chromosomal segment does not happen in the if crossing over happens in the paracentric inverted segment. Okay, so this is the reason why I took the question, so that you understand how to take call in this kind of questions. Okay, right. Now before I go ahead, so this is the advertisement from Biotechnica. We are opening our Josh batch, and this will be coaching given for December as well as June. Okay, 1 year covered, 1 year of your CSIR net preparation covered. So it's a double success guarantee batch that we are starting. And as you know, Biotechnica does not offer just coaching classes, it offers so many different freebies along with your course. Okay, so everything is there. Batch starts from 1st of July, but you have to understand that there are limited seats available. Okay, so all the information is given on the screen. If you want to note down, please note it down and give us a call if you are interested for Biotechnica's enrollment. Right. Moving on to another question. Now, this is also a type of question which is kind of frequently asked in the CSIR exam, where they will combine different concepts. Okay, and the concepts which are combined here are pedigree analysis as well as the molecular markers. Okay, so very frequently this is like in a question in a sessions question paper, two or three part C questions you will see where the marker related questions are there. Okay, so molecular markers, if you guys are thinking of skipping it, that will be very very wrong. Please do not skip it and prepare molecular markers, because molecular markers are something that can be combined with many different topics. Okay, and that is getting combined. A lot of questions are coming from molecular markers. Right, so let's have a look here. So this is a hypothetical example. Pedigree of a monogenetic trait is given below. The shaded individual shows spotted skin color while the rest have uniform skin color. The individuals 1 to 9 in the pedigree were analyzed for a DNA marker, both fragments that shows complete linkage with the skin color trait. Okay, the following statements were made regarding the above observation. Okay, so it says that statement A, spotted skin color is a dominant phenotype. Right, now if you see the pedigree chart, okay, so what will be your first understanding? That this is a recessive trait. Why? Because 3 and 4 both are not affected, but they have children who are affected. Okay, and 1 is affected, 3 who is daughter of 1 is not affected, and again 6 7 are affected. So this is typically a skipping of generation, which is not shown by dominant traits. Right, so your first instinct will be to go for recessive trait, but because there are two charts given, so we have to analyze this also. You cannot just answer by analyzing the pedigree chart, you have to look at the markers also. Okay, now see markers. What is the benefit of molecular markers? That even though the individual is showing the trait or not, if you have found a molecular marker which is linked with that particular trait, so the molecular marker will show the pattern. Okay, and if that pattern is shown, now irrespective of whether the symptoms are there or not, you can say that this individual has that particular trait. Okay, so even though the phenotype is not there, but the causative gene is present. Okay, so for that what we have to do is see 1 is affected, to see the pattern which is shown by 1. Okay, this means 2 bands are there in 1. Okay, now 6 and 7 are also affected. Now for 6 and 7 also we see that there are 2 bands. Right, so what can you say from this information? That this double bands, who all will be having the double bands? The one who have this spotted skin color. Okay, or they have the gene for skin color, the spotted skin color. Okay, now along with this 1 6 and 7 who are also affected in the pedigree chart, so we see 3 and 5 who also have this double band. Okay, so since they have the double band, what does it mean? That they also have the allele which can produce the spotted skin color. Okay, and who is 3? 3 is mother. Right, so if 3 is mother, and through 3 only it is passing on to 6 and 7. Right, so with what does it mean? So every affected individual has one parent who is carrying the causative allele. Okay, so this is a dominant trait. So spotted skin color for sure is a dominant phenotype. Okay, now your question, so if 3 is having the trait, like the causative allele, then why is she not showing the phenotype? So answer will be because of partial because of so because of the variable expression. Right, so spotted skin color, so this statement A is correct. So B says that spotted skin color shows variable expressivity. Now it's not expressivity, because expressivity would have been that some of them will have lesser spots, some of them will have more spots. Okay, here what is happening, that some of them are not even showing the phenotype even though they have the causative value. Okay, so that is penetrance. right? So this three individual three is not showing the phenotype because of partial penetrance. Okay, because penetrance is not 100% for this particular allele. Okay, so it is not expressivity, it is the partial penetrance which is causing this situation. So B is incorrect. Statement C, the DNA marker associated with the skin color trait is codominant. Yes, that is also true. Okay, how can we say that? Because codominant markers, they show a different like they will be present in heterozygous condition also, right? They will show a different pattern in the heterozygotes, and they will show a different pattern in the homozygotes, right? So here, as we can see that the individual, so if the individual one, okay, so what will be her genotype basically? So if we say that this is a gene A which is causing, capital A is dominant and the causative allele, and small a is recessive. So basically the genotype of one will be capital A, small a. Okay, so they have a separate band pattern, heterozygotes. So what will be the what will be the genotype of two? Be small a, small a. Okay, so homozygotes have a band pattern which is different from the heterozygotes because the marker that is there is a codominant marker. Okay, right? Now D says that the probability that the individual 5 will pass on the allele responsible to the spotted skin color to the next generation is 0.25. Okay, now here it is not 0.25. So first of all, what will be the genotype of 5? So 5 also has this two bands, right? Means 5 genotype will also be capital A, small a. Okay, now provided 5's wife, sorry, 5's husband is not affected or does not even have the positive allele, in that case cross will be capital A, small a with small a, small a. Okay, and in that case what will be the chance of having capital A dash? Will be 1 by 2. Okay, so there will be 0.5% 0.5 will be the probability of passing, not 0.25. Okay, so statement D is incorrect. So answer will be A and C. So guys, I would like to remind you all again. Yes, that's correct, Akshata. This was an example of incomplete penetrance. Okay, next question. Now this is also a question which is getting asked very very frequently, and again, as you can see, it was asked in December 2023. There will be a similar kind of question in June 2023 also, December 2022 also, June 2022 also. Okay, so transposons are becoming one of the favorite segments from where questions are coming, and this part is can be considered as part of unit 2 as well as unit 8. Okay, so the statement below attempt to describe a few characteristic of ALU repeat which is found in the human gene. So ALU elements are a class of short intersped elements. That is true. Okay, so ALU is an example of sign, right? Now signs are autonomous transposon is wrong. Signs are short interspersed nuclear elements. So signs basically how they are short or why they are short, because they do not have the gene coding for transposase, or they do not have the gene which will be coding for the enzyme that will help in its transposition. Okay, so signs are non-autonomous. So signs will transpose using the autonomous transposon. So lines in this case would have been the autonomous. Okay, so signs are non-autonomous. Statement B incorrect. Statement C, okay, ALU repeat originated from cDNA copies of 7 sLRNA. D says that ALU repeats have a relatively high AT content. E says that they are preferentially located in the gene poor G chromosome bands. Now, if you know only this much information about sign, okay, this is so funny, I'll tell you that if you know about that ALU element is sign and sign is non-autonomous. Okay, with this much information also, because these are more detailed information, but with this basic information that ALU elements are signs and signs are non-autonomous, you can solve this question. Because what do you know? That statement A is correct and B is incorrect. Okay, now scan the options. So you have which all options has A. So you have 1 and 3 which has A. Okay. Now you know that B is incorrect. So you have to eliminate out 1. So you got your answer, option 3. Do you see, guys? So it's just like the first question, that if you solve the easier part, you can get the answer. Okay, so the difficult part is kept, definitely it is there, but it is also there to check your aptitude. Okay, the basic general aptitude, that how good are you in analysis. Okay, and here also you can see that even if you do not know this detailed information about the ALU segment, still you can answer this question this easily, and actually less than 30 seconds you can solve it. Right? So answer will be A and C. And C says that ALU repeat originated from the cDNA copies of the 7SL RNA. Okay, so this is an extra information that you can keep. All right. Next question from June 2023. So red blue color blindness is a human X-linked recessive disorder. The two parents with normal color vision have two sons. Son 1 has 47 XXY chromosome composition and is colorblind. Son 2 has 46 and XY composition and is also colorblind. Okay, so both the sons are colorblind. Assuming that no crossing over took place in prophase 1 of meiosis, Klinefelter syndrome in son 1 resulted due to the non-disjunction during which one of the following events? Okay, so the two parents with normal color vision have two sons. Okay, so both the parents have normal color vision. Now both the sons, as you see, are colorblind. Okay, now color blindness is also mentioned that it is X-linked recessive disorder. Now this color blindness is such a popular disorder that you should know also that it is X-linked recessive. Okay, now if it is X-linked recessive, what is the inheritance pattern of X chromosome? That it will show a crisscross pattern of inheritance, right? So basically what will happen, that this is male and this is female. Okay, so females will pass their X chromosome to both son and daughter. Okay, but the sons will get their Y chromosome from their father, and X chromosome from the father will go only to daughters. Okay, so X chromosomes basically show a crisscross pattern of inheritance. So sons will receive their X chromosome only from mother. Sons will get X chromosome only from mother. Okay, because from father they will receive their Y chromosome, right? So if the sons are getting it from mother, that means mother has to be the carrier. Okay, both the parents were having normal vision, so if both the sons are getting color blindness, that means mother was the carrier, right? So here it is saying that non-disjunction happened in which one? So male gamete you can completely eliminate, because father doesn't even have the causative allele, right? So only mother has it. Okay, now it is saying that in which meiosis. Now here you have to, it has to be very clear to you that what kind of chromosomes separate during which meiosis. So during meiosis 1, it is the homologous chromosomes that separate. Whereas in meiosis 2, it is the sister chromatids that will separate. Okay, now in the son 1, who is also having the Klinefelter syndrome, he has color blindness. Okay, so if he has color blindness, so if I say that this color blindness is because of this XCLE, okay, which is recessive, that means this son 1 has the genotype of XCXCY. Okay, because he has color blindness, that means both the X chromosome has the XC. Okay, so now mother was heterozygous. Okay, so what was mother's genotype? Mother's genotype, in this case, the mother's genotype was XXC, right? So if XXC is there and in son 1, both XC chromosomes are coming, means what? The sister chromatids are not getting separated, right? Because if homologous chromosomes are not getting separated, but homologous chromosomes have different alleles, right? One has the normal allele, one has the color blindness allele, right? So if both the X chromosomes that are received in Sun1 are having the XC allele, means there is non-disjunction happening during the meiosis 2, where the sister chromatids are not getting separated. Okay, so answer will be option B. So, guys, we are kind of halfway through the sessions. I would definitely like to have a review from you all. So, how is it, guys? I would like to have a feedback. Yes, how is it? Are you enjoying the session? Are you finding it helpful? These questions, are you able to solve them on your own also? Yes, can I have some quick responses, everyone? Okay, Akshata. So I hope you are doing your practice and you all have started solving questions, all have started taking timed mock tests. Okay. Next question. Now, this is also a type of question which can be answered very, very easily. Okay, less than 30 seconds, provided you have the underlying concept of it. Okay, and this concept is not related to pedigree. Here it is a combination. Okay, so as you see this question, the pedigree below is a reference to Angelman syndrome, which is caused by a mutation in UBE3A gene on chromosome 15. The gene is also paternally imprinted. Individuals showing AS have not been indicated in the given pedigree. Individual 1 does not have AS. Individuals marked with dots are carrier of this particular mutation. Which one of the following option least individual, all of whom are likely to show AS? Okay. Now, for this, you have to have this understanding that Angelman syndrome and Prader-Willi syndrome, they both happen because of mutation in the same region of chromosome number 15. Okay. But the segments are, along with they causing this kind of syndromes, these two different syndromes, they also are parentally imprinted. Okay. Now, because of parental imprinting, the segments become non-functional. Okay, what is imprinting? Imprinting means segment of genes or segment of a chromosome is tagged for getting silenced or for not getting expressed. Okay, now this is something which is a epigenetic control which happens in the different segments of our genome. Okay, so this segment of chromosome number 15 is also under this parental imprinting. Okay, now, so because of that, what happens that if you inherit this particular mutation from mother, so I'll just write it here. So if this mutation is inherited from mother, then it causes Angelman syndrome. Okay, but if the same mutation is inherited from father, then it causes Prader-Willi syndrome. Okay, so with this much information, you can solve this question this fast. How? Because what we have to see, you have to check the individuals who will show this Angelman syndrome. Sorry. And it is already mentioned who all have this particular mutation. So every individual who are having this dot in them, they are having the mutation. Okay, so who all will show Angelman syndrome? The ones who are getting the mutation from their mother. Okay, so as you can see, 2 and 2 5, they are inheriting it from father, so they will not have Angelman syndrome. In fact, they will have Prader-Willi syndrome. Okay, but that is not asked in the question. 3 1 and 3 2, yes, they are getting from mother. Okay, so they both will have Angelman syndrome. Again, 3 5 will not have. Okay, but 4 2, is getting it from her mother, so she will also have Angelman syndrome. Okay, so who all will be having Angelman syndrome? 3-1, 3-2, and 4-2. So answer is option B. Okay, so guys, this kind of questions are extremely easy. Only thing that they require is the topics or the concepts that they are combining in the question. Okay, when you know that, you can solve this question so fast. Okay, moving on to a question from microbial genetics. So, as you know, yes, I tell you repeatedly in the class also that microbial genetics, yes, interrupted mating experiment is one of the segment from where questions are asked very frequently. So, in this question, what is given? 4 different HFR strains of E. coli were mated with F minus recipients, and the time of entry of various donor markers were found to be as below. So, we have HFR1, and the information is given for 2, 3, and 4. Based upon the above observation, the following statements were made assuming MET to be 0 minute and 3O at 15 minutes. Okay, so now these are statements given A, B, C, D. Which of the following options represent all correct statements? Okay, so for this question, for you to solve this question, what you have to do? You first have to make the gene map. Yes, shall I give you guys 2 minutes to make the gene map? Yes. Yes, tell me. Will you like to make the gene map? Come on, participants, just send some message to me because I don't know whether you guys are going to make it or not. Do you all know how to make microbial gene map? Where you can make a circle and put the markers on it. Okay, Akshata, very good. What about others? Nobody else is doing it. Okay, so let's make the gene map, everyone. So for gene map, what you have to do is, first of all, you have to make a circle. Those of you who do not know how to make bacterial gene map, pay attention now. So first of all, bacteria has circular DNA, so we will make a circle. Okay. Now, what is the concept of interrupted mating experiment? That we will stop the mating at different time intervals. So whatever gene has transferred, that is how the genes are placed one after another on the chromosome. Okay. And also by looking at the time lapsed between the transfer of two genes, we can also find out the distance between them. Okay, now all of this calculation is based on that MET is passed at 0 and 3O passes at 15. Okay, so what we will do, we will find out the gene, the distance between them. Okay, so for HFR1, MET is going first, so let's just put MET here. Okay, after MET there is 3O. Okay, then there is PHE, then there is MAL. Okay, now what is the time lapse between each of them? So 3O is going at 30 minutes, met at 15. So distance between them will be 15 minutes. 30 minus 15, right? So here it is 15 minutes. For 3O and Phi, it will be 42 minus 30. So 12. Okay, and then it will be 57 minus 42, right? So it will be 15 between Phi and Mal. Okay, so I hope all of you understood how to make the gene map. So this is how you can put all the markers. Okay, so now in for the HFR2, what you will do, we will look for the overlaps. Okay, so where are the overlap is here at mal. Okay, so on one side of mal, if there is phi, on the other side there will be his, right? So after mal there will be his here on the other side. And what will be the distance between mal and his? So it will be 77 minus 60. So 17. Okay, like that you can put the other gene markers. This will be thigh and then bio. Okay, and this is how you can find out the distance also between them. Okay, so his and between his and thigh, what it will be, 60 minus 51, so 9. Okay, and thigh and bio, so you see there is a difference of only 1 minute. So actually let's I should put bio very close to thigh because distance is only 1 minute. Okay, so likewise you have to put the rest of the gene markers and find the distance between them. Okay, so I have already done it here. So this is how your gene map should look. Okay, where all the markers which are mentioned in the question, we have placed it on the gene map and we have also calculated the distance between them. Okay, now once that is done, now we have to check for the statements which are given in the question. Okay, so if we look at the statements, it says that first one, his is located at 59 minutes. Okay, so we have to start from met, remember. So distance between met and his, how much it will be? So we just have to add the numbers, right? So it will be, so met to 3 is already 15, so 15 plus 12, that is 302 phi, then phi to mal plus 15, and then mal to his 17. Okay, so this is coming as 59 minutes. So this statement is correct. Let's check the second statement, which says that Azai is located at 74 minutes. Okay, so till his we have already calculated, that is from met to his we know is 59. Okay, so we don't have to add them all again. We can just continue from here. So met to Azai will be 59 plus 9 plus 1 plus 5. Okay, so how much it is? Yes, 74 it was given. Yeah, and this is coming as 74. Okay, I hope all of you are getting it. Now, third statement, cis is located at 11 minutes. Okay, so for cis, right? Now, cis, met to cis's distance is not given. What is given is met to 3O and 3O to cis, which is 4. Okay, so from the diagram itself, you can see what will be the distance between met and cis. It will be 15 minus 4. Okay, so it will be 11 minutes, which is also true. Now, what about the last statement? Mal is located at 76 minutes. So if I say met to mal, okay, where is mal? Here. That is, it will be 15 plus 12 plus 15 is equal to 42. Okay, but it is saying 74, right? 76. So that will be wrong. So this statement 1, A, B, and C are correct. D is wrong. Okay, so I agree this question is little lengthy because first you have to make the gene map and then do the calculation, but it is not difficult. Okay, plus minus only you have to do. No other mathematical skill is involved. Okay, a quick reminder for our Josh batch. Anybody who missed and the this advertisement and whoever has joined new. So Biotechnica is launching Josh batch very soon, starting from 1st of July. There are limited seats available, and you get all these benefits, which I cannot even say everything here because we will be running out of our session. So all the information is given on the screen. If you need further information, please give us a call in the toll free number, or you can visit stores.biotechnica.org. Next question, which is a very popular kind of question, which is again has been asked multiple times, okay, multiple times from CSIR NET exam, okay, where and this type of question is based on mutation reversal study, okay. So scientists synthesized 4 new chemicals which had mutagenic potential and named them as C1, C2, C3, and C4. He tried to analyze the nature of this mutation caused by them and obtain the following results. Which of the following answers describes the mutagenic potential of the chemicals? Okay, so in this case, what do we have? So C1 is getting reversed by 2-aminopurine, nitrous acid, hydroxylamine sum, acridine orange. No. Okay. So first of all, whichever known chemicals we have used, so we have to associate them with the type of mutation that they can cause. So 2-aminopurine is a base analog. Nitrous acid is a deaminating agent. Hydroxylamine is a hydroxylating agent. And all three of them causes transition-based substitution. Okay. Acridine orange is a intercalating agent and it results into insertion or deletions. Small insertion or deletion. Okay. This also is important. Right. So if C1 is getting reversed by 2-aminopurine and nitrous acid, that means what? That C1 has to be a transition. Okay. Now C2 is not getting reversed by any. That means C2 is neither transition nor small insertion or deletion. Okay. Apart from that, we can't say anything. Okay. So we'll leave it at that. C3 is again getting reversed by this 2. So again, so yeah, now this 2 can only do transition. So C3 has to be a transition all again. Okay. So C3 is also a transition. And C4 is getting reversed by acridine orange, means C4 will be a small insertion or deletion mutation. Okay, now let's scan the options. So C1 has to be transition, right? So C1 transition. Now with this only you have got your answer that C1 transition is there only in option A. B says that is transversion, wrong. C says that large deletion, wrong. And here it says that transversion, again wrong. Okay, so you see only one thing you have to solve and you have solved the entire question. Okay, so that is the smartest way of approaching CSIR question paper, guys, that you just solve half of the question, okay, and then scan through the options, okay, or first only scan through the option, so it will give you an idea in which direction you have to think. Okay, that is the benefit of multiple choice questions. Okay, and in those kind of questions where you are not completely sure about the answer, you can go by the sequential elimination process. So start eliminating the options which are completely incorrect, okay, and then slowly slowly go to the option which seems somewhat correct. So C1 is transition, C2 is transversion. Now that can be true because transversion is neither transition or a small insertion deletion. So that is can be correct. Or a large deletion is also true, possible. C3 is transition, right? And C4 is single base insertion or deletion. So answer is option A. Next question, complementation test question, which is again gaining its popularity. Every session nowadays question are coming from complementation test. Either they will give you the regular table of a complementation test with plus and minus showing complementation and no complementation, or question will be like this where it is not directly mentioned that there is complementation test involved, but seeing the question you have to conclude that. So question says that Drosophila mutant line A with vestigial wing is isolated in a laboratory. The vestigial wing phenotype was observed to be recessive and mapped to gene X. Three other laboratories also isolated vestigial mutants called line B, C, and D. In order to test if the mutations in line B and D is also mapped to gene X, the following crosses were made and phenotype of F1 progeny observed. Okay, see, where is the clue that it is complementation test? So first of all, what it is saying, in order to test if B and D are also mapped to gene X. Okay, complementation test, is it not what we do to find out whether the mutations are happening in the same gene or different gene? Correct. And also what we are doing this cross and then we are checking for the phenotype of F1. Excuse me. So this is something that we do in the complementation test. Okay, so this is basically a result of complementation test given. So based on the above, identify the lines which are most likely not to have mutation in gene X. Okay, so here what is the basic understanding that you need? That the genes which complement each other, okay, so they will produce the wild type phenotype. Okay, so wild type phenotype in progeny. Okay, so F1 having wild type phenotype means complementation has occurred. Okay, and if there is complementation means mutations are in different genes. Okay, so what will be the counterpart of this? That if F1 is mutant, then there is no complementation has happened. And when will this scenario be? When the mutations are in the same gene. Okay, now question is which of the mutation is not to have in gene X. Okay, it means we have to find out the mutations which are not having the which are complementing each other. Okay, so now normal is wild type and vestigial is mutant. Okay, because mutant line A produces vestigial. So vestigial is mutant, normal is wild type. Okay, so AB vestigial, that means in terms of complementation, what it means? No complementation. AC vestigial, no complementation. AD normal means there is complementation. BC again vestigial, no complementation. BD complementation, CD complementation. Okay, now if you have to put the genes together, so A, B, and C are not complementing each other, means they belong to the same gene. Okay, but D is complementing A, D is complementing B, D is complementing C, means D is present in a different gene. So if A is present in gene X, means B and C are also present in gene X. Okay. Yes, absolutely correct, Akshata. So A, B, C will be the allelic mutations and D is present in a different gene. So which line is not having mutation in gene X will be line D only. So the concept of complementation test is also very, very easy, guys. And please do not skip that as well because questions are coming repeatedly from complementation test. Okay, now this brings us to the very last question of the session. Here we have, this is from polygenic inheritance and it is a very smart question. From polygenic inheritance, yes, there can be numericals which can be asked, but nowadays we see that questions are coming more theory based from heritability, okay, from the concept of heritability or also from the concept of QTL mapping, which can also be linked with unit 12. Okay, but I found this question very interesting because it has the concept of heritability. Along with that, there is a calculation also which is involved. Okay, so let's have a look at this question. This was asked in CSIR June 2017. Two inbred lines of beans were intercrossed. In F1, the variance in bean length was measured as 2. The F1 was self to obtain F2 and the variance in bean length in F2 was 7. The broad heritability of bean length in F2 population will be. Okay. Now, first of all, what is broad heritability or broad sense heritability? Now, broad sense heritability. So, first of all, heritability. So, heritability gives us an information about what proportion of the variation in the phenotype that we see in the population is because of genetic cause and what proportion is because of environmental cause. Okay, so broad sense heritability, which is represented as H2, it shows the it shows what variance in the population is is because of the genetic variance. Okay, so in mathematical terms, if we see, so it is equal to Vg by Vp. Okay, so Vg is the genetic variance, Vp is the total phenotypic variance. Okay, now now from where we will get this Vg and Vp, right? So there are some numbers given. Now what are they? Let's see. So F1 variance in bean length was measured as 2. Okay, so see, there are two inbred lines of beans were crossed. Two inbred lines means they were purebred or they were homozygous. So if they are homozygous, what do we expect? F1 to be heterozygous and F1 will have only one genotype, right? Whether you are considering one gene or multiple gene does not matter. If you have crossed two inbred lines, F1 will have only one genotype, right? So when they all have one genotype, ideally they should all have one phenotype. Okay, but still we see that there is a variation. Okay, why this variation is there? Because of environmental factor. Okay, so whatever variation that we are seeing in F1 is not because of the genes but because of environment alone. Okay, so what is this 2? It is VE. Okay, so variance because of environment is equal to 2, right? Now in F1 generation, when it is selfed, we get F2 and the variance in bean length in F2 was 7. Okay, now in F2, there will be different genotypes and there will be different phenotypes. Okay, but when we are talking about the expression pattern of a particular gene, so environment also plays a role. So basically, the phenotype produced by a particular genotype will be an addition of the genetic effect plus the environmental effect. Okay, so what is this total bean length variation in F2, which is equal to actually Vp? Okay, and this Vp, it is equal to Vg plus Ve. Okay, now Vp is how much given? 7. Ve we are already getting from F1 is equal to 2. Okay, so how much will be Vg? So Vg, that is the variance, the genetic variance will be equal to 7 minus 2, that is 5. Right? So now we have got Vp, value of Vp also. Vp is equal to 7 and Vg also, which is equal to 5. So how much will be the broad sense heritability? It will be equal to 5 divided by 7. Okay, so that will give us 0.71. All right. See, AAA, yes. So we have three last minutes. Yes. So AAA, when we are talking about production of a genotype, sorry, production of a phenotype. So earlier the concept was that whatever is the genotype, it will produce the associated phenotype. Okay, something that Mendel worked on. Okay, but with further information and more research, scientists have found out that genotype is not the sole cause of producing phenotype. Genotype is also worked upon by environment and then only the phenotype is produced. Okay, so whatever variation that we see in the phenotype will be the variation because of genotype plus the variation because of environment. Okay, so that is why we get this formula Vp, which is phenotypic variance, is equal to Vg plus Ve. Okay, so because F1 there is no genetic variation, only environmental variation is there. So 2, whatever the variance that we are getting is equal to Ve. Okay, now in V2, in F2, sorry, there will be genotypic as well as environmental variation. So we already knew the value of Ve. So from there we calculated out what is Vg and then we substituted in the formula for broad sense heritability and we got this. Okay, so guys, if you want to If you want to know a little bit more on this, so your basic concept of broad sense heritability or this environment and genotype reducing phenotype has to be clear. Okay, so just go through it once again and see this recording once again, and you will understand the concept. Okay, so yeah, so the solution was given here. All right, so guys, many of life's failure are people who did not realize how close they were to success when they gave up. Okay, that was said by Thomas Edison. And this is not the time to give up, this is the time to charge. Even like you have to put your booster force on and start practicing and revising on your full force, and you will succeed. All the best, everyone. Thank you everybody for joining the session. Thank you, everyone.